In this lecture, let us look at the temporal dynamics of functional MRI. As I had mentioned in one of my previous lectures, the way we measure brain function using MRI is by observing the changes in the hemodynamics. Now these changes do take some time. So for example, on the left here, you can see the time course during a 30-second period of finger tapping. And you can see that the signal slowly increases, it reaches a plateau, and then when the activity stops, the signal slowly returns back down to baseline. And similarly, if a person were to tap only the finger for 1 second, the signal slowly increases, reaches a peak at about 5 seconds, and then slowly decreases down, with the entire hemodynamic response lasting about 10 to 15 seconds. Note also this post-stimulus undershoot here. What could be causing that post-stimulus undershoot? Well, one clue comes from a study by Joe Mandeville, where they were looking at changes in BOLD signal as well as cerebral blood flow and cerebral blood volume, or CBV. And what they noticed was that the CBV was elevated after the task was done and was more slowly returning to baseline than both the BOLD signal and the cerebral blood flow. This led to the development of the balloon model. So with this model, as shown here in a schematic of a blood vessel with the different blood cells, red indicating oxygenated blood cells and purple indicating the deoxygenated red blood cells. What happens when a neuron becomes more activated is at first, perhaps there's an increase in the oxygen extraction. This initial dip actually is not very commonly observed in fMRI. However, what happens soon after that is that the blood flow velocity increases, and this causes the increase in the MRI signal as deoxygenated blood is being pushed out. In addition, this increase in blood flow causes the veins and venules to dilate, resulting in an increase in cerebral blood volume. Now, after the task or stimulation is done, the flow returns to normal. However, the blood volume, as you saw in the previous graph, is more slow to return to normal. So as a result, we have an increase in blood volume with the same flow as we had initially, resulting in more deoxyhemoglobin being present in that voxel. This causes a signal decrease below the initial baseline. And then finally, as the volume returns to normal, the signal returns to its normal baseline. Another question that was asked early in fMRI is whether we can detect the BOLD response to brief stimuli. And indeed we can. Here's an example showing the visual stimulation to visual activation to a 1 second visual stimulation, 100 millisecond and 34 millisecond stimulation. And we can see a BOLD response even to the very brief stimulations. Another question that was important to address early on in fMRI was whether the BOLD response was linear. In order to do that, this is some work from Jeff Boynton. They presented a stimulus, either a single brief period, 1 second period of flashing checkerboard, or repeating that twice, 0 and 2 seconds, or repeating that stimulus 3 times. If you look at the responses from that, here you can see the responses from 1 trial, from 2 trials, and from the 3 trials. Now, if you subtract the response from the 1 trial, if you subtract that from the response to the 2 trials, what you end up with is as shown here. And similarly, if you subtract the response to the 2 trials from the response to the 3 trials, you end up with another curve. And you can see that these curves generally overlap pretty well, suggesting that really the difference between the 1 trial and the 2 trial is the same as just adding another one of these 1 trial responses. So in general, the BOLD response is pretty linear. And this is good news if we want to use things like general linear model in order to model our BOLD response. Jeff Boynton also noted in this particular study that the BOLD response did appear to be somewhat nonlinear for very brief stimuli. That is, when the stimuli were less than a second in duration. What's interesting about that to me is that for years after that, whenever somebody wanted to cite a paper that showed that the BOLD response was linear, they cited his paper. And similarly, when they wanted to cite a paper that showed that the BOLD response was nonlinear, they also cited his paper. So this kind of goes to show that it's good to be very thorough in reporting your results. Another important aspect of the temporal dynamics of the fMRI signal is the variations in the latency of this response. So what I'm showing on the left is the activation amplitude, and below it the delay in that response, that is the latency of that fMRI signal change. And in general, we see some hotspots in terms of the activation amplitude, and these typically correspond to larger vessels. And you also see some hotspots in the delay. But importantly, note the range here. This is plus minus 2 seconds. This is much larger than anything we would expect based on actual differences in the neuronal activity. A histogram of these differences is shown on the right. These differences are largely due to what kind of blood vessels are in that particular voxel, with the larger veins being downstream and showing more delayed signal changes than the capillaries. So because of this, it's very difficult to determine whether one brain region fired before another brain region using fMRI, because we have this large range in latencies. Now, there are some ways around this, and this was demonstrated in this rather clever experiment, also from Peter Vanitini. Now, in general, your left visual field is mapped to the right hemisphere, and the right visual field, what you see on the right here, is mapped to the left hemisphere. And in this experiment, he first had a fixation point, and then he showed these contrast reversing dartboards, but he turned on the left visual field just a few hundred milliseconds before the right one, and then turned it off a few hundred milliseconds before the right one as well. So now if we map the latencies, in general, you would expect the latencies in one hemi-field to be more delayed than another. So if you turn on the right and then the left, here's the latency map you get. Really doesn't very clearly show differences in the right versus left hemispheres. However, what we can do in the this visual experiment is flip the stimuli. So instead of turning on the right and then the left, let's turn on the left and then the right. So again, if you map the latencies, this is the latency map you end up with. But now what happens when we subtract the two? We exactly see this difference in the left versus right hemispheres. So the trick here is, instead of relying on the absolute latencies of the fMRI response, instead we're looking at the relative latencies. That is, we're looking at how the latency changed when we've changed our stimulus. Or in other words, we've subtracted out the hemodynamic latencies in doing the subtraction, and what we're left with is the changes that are due to the modulation of the task, the differences in neuronal activity. Now, this can be done not just in the visual cortex. Here's an example from a study from Pat Belgowan showing a task where a person had to decide whether something was a real word or not. That is a word or a non-word. That is a word that is pronounceable but is not a real word in the English language. And these words were also rotated by different degrees. Now, in order to do this task, your brain first has to mentally unrotate the word and then decide whether it's a word or not. And they could see in different parts of the brain that in some cases there were areas where the response was more prolonged, and in other brain areas there were areas where the brain response was more delayed. And so based on this, they could tell what brain areas were actually firing before others and how this information flowed. So the trick really here is that we're not relying on the absolute latency, but rather we're looking at the relative latency. That is, how is the latency of the fMRI response changing when we change a particular task or stimulus? Another way we can take advantage of the slow, bold hemodynamic response function is in this example from actually some of my PhD thesis work. And that is one of the challenges when we want to have a person, say, speaking inside the MRI scanner is that they also move their head a little bit, or even the movement of the jaw and tongue during the speech can produce artifacts. So this is an example here. We see some motor cortex activation during this periods of speech. But you also see a lot of signal changes at the edges of the brain indicating some motion artifact. However, if instead of speaking for, say, 20 or 30 seconds at a time, alternated with resting periods, as like this block design, if we have the person speak just a single word and then wait, the artifact, the movement happens right during that speech, whereas the bold response is delayed and happens after that. So if we map that out, you can see a much cleaner activation map without the artifacts at the edges. Here's another example of that, just a different particular study, again showing how you can reduce the artifacts by going to more event-related design. And in fact, there's a whole set of different kinds of timings you can come up with where the correlation between the expected bold response and an expected motion response, which is exactly synchronous with the task, would be relatively uncorrelated. And I'm going to draw your attention in particular to one of these, that is the top row of these low correlation with motion, and that is a task with 10 seconds of task alternated with 10 seconds of rest. And in this particular design, the BOLD response is exactly shifted by a quarter of a cycle so that it's uncorrelated with the actual task design. And if the motion is correlated with the task design, that means any motion-related signal changes should not be correlated with the expected BOLD response. And that's what we demonstrate here. This was actually having a particular person doing this block design task of only 10 seconds. In this case, this was a verbal fluency task. And you can see that in the motor cortex, where we expect the BOLD response to be, we get exactly the response at that latency, whereas in other areas, like down in the area of the mouth and the jaw, or in the inferior temporal cortex, we see much earlier BOLD responses than we'd expect due to an actual motion artifact. And we then use this particular task in studying the verbal fluency. And by doing that, we were looking at the differences during this task in individuals with autism spectrum disorder versus typically developing controls. And we found significant differences in a region of the fusiform gyrus, the visual word form area, where in comparison, we did not see differences in the actual production areas where they're actually trying to produce these words. Finally, another important feature about the temporal dynamics to keep in mind is related to how slow the signal changes we can detect. And the fact that with BOLD fMRI, it's very difficult to detect very slow changes. In this particular study, they were comparing perfusion imaging to BOLD imaging. Let me draw your attention in particular to this optimized BOLD imaging column. So here you see the activation in alternating, in this case, 30-second periods of activation with 30-second periods of rest. In this case, 1 minute periods of finger tapping, alternate with 1 minute periods of rest, or 2 minutes, or 4 minutes. And you notice as you get to these very long periods, 4 minutes of finger tapping versus rest, we lose really the signal. The reason for that is that there's a lot of noise in our fMRI data at these very low frequencies, or in other words, there's a lot of drifts in the data. And so it can become very difficult to distinguish these very slow true signal changes from all of the surrounding noise. So this is important to keep in mind when you're designing your fMRI study, that it is very difficult to detect these very slow